Today, I will be discussing part 1 of cell division. The topics include mitosis, which has the prophase, metaphase, anaphase, and telophase, parts of the chromosome, phases of the cell cycle, and regulation and timing. To start, we have mitosis, which functions in growth and repair. It produces two genetically identical daughter cells. Chromosome number 2N remains the same, and it consists of phases prophase, metaphase, anaphase, and telophase. To start, we have to be able to identify the parts of the chromosome. The kinetochore is a disc-shaped protein that attaches chromatid to the mitotic spindle. The centromere is a specialized region that holds two sister chromatids together. The sister chromatids are two strands of DNA. One is a replica of the other. Next up, we have the phases of the cell cycle. The interphase consists of G1, S, and G2. G1 consists of intense growth and biochemical activity. S consists of synthesis or replication of DNA. And G2 consists of cell, continues to grow and prepare for division. Most of a cell's life is spent in the interphase. The nucleus is visible. Next, we have mitosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm. In order for a cell to function, it has to have regulation and timing. Two important factors limit cell size and promote cell division: the ratio of the volume to the surface area and the capacity of the nucleus. There are built-in checkpoints at G1, G2, and M that either halt or trigger cell division. The most important checkpoint is the restriction point in G1. Without the go-ahead signal, the cell becomes a non-dividing cell. For example, muscle and nerve cells. Timing of cell division is also controlled by cyclins and cyclin-dependent kinases. Cyclins are synthesized in response to various molecular signals, including growth factors. To start, we have the first part of mitosis, which is the prophase. It's where the nuclear membrane disintegrates or disappears. Strands of chromosomes condense and become visible, and then the nucleolus disappears. Next up, we have mitosis metaphase, which is where chromosomes line up in a single file on the metaphase plate. Centrioles are at opposite ends of the cell, and spindle fibers run from centrioles to kinetochores. Next up, we have mitosis anaphase. Sister chromatids separate as spindle fibers pull them apart. This is the shortest phase of mitosis. Lastly, we have mitosis telophase. This is where chromosomes cluster at opposite ends of the cell. The nuclear membrane reforms. Supercoiled chromosomes unravel and become visible again, and spindle fibers may still be visible.